in this video we focus on the tools package available in the offline DHA and DLHA modules. To explain the filter sections, I have already reconstructed a hologram so we can use its phase and amplitude images in the next examples. So in this area, you can apply filters to the hologram image, the phase, or the amplitude. There are 4 filters you can use with a slider to set its level, and there is one adapted filter with a simple checkbox to turn it on or off. To use a filter, first select which image you want to affect, phase or amplitude, or maybe the hologram. Then pick the filter, set its level with the slider and press apply. The gamma filter changes how light and dark values are spread, so it can make the image darker or brighter. The contrast filter pushes tones away from or toward the mid-level. So higher values increase contrast and lower values reduce it. The higher pass filter removes a smooth background and boosts edges and fine details, which is useful for seeing contours. The low pass filter smooths the image to reduce grain and noise, but it can also remove some details. And the adaptive filter redistributes intensities to use the full dynamic range. It often reveals hidden details, but it can raise noise in flat regions. And as we have seen, if you want to remove a filter, just unselect it and press apply again. This filters area also includes visualization color mode. Here you choose the color map for how the phase or amplitude is shown. Select the image, pick the color map you want, and click apply. If you want to go back to the original look, choose original and press apply. And that's all for the filter section.
Now we will explore the bioanalysis tools integrated into the offline DHN mode. We will work with a real biological sample. Let's begin by loading a hologram of a red blood cells from main menu, load and load the hologram. First, we compensate the hologram and refocus the sample. Now we open Tools by Analysis. Here we have 3 panels: Dimensions, QPI measurements, and macrostructures metrics. Starting with Dimensions tool, we select what image we want to analyze. Then insert the pixel size and the optical magnification. Click the Apply button. Then click 2 points on the image corresponding to opposite edges of a single cell. The measurement distance appears in the text close to the cell. The same with the scale bar. You can change the position of the scale around the image. If you do double click, you can change the length of the scale. And if you do right click in the scale bar, you can change the color. Next, let's move to the QPI measurements tool. First, select the linear row mode, then insert the number of zones you want to measure, then index of the sample and the median. Draw a straight line across on red block cells, passing through its center. The software immediately plots the face profiles along that line. Below the plot, a table appears with the information of the face and the thickness. Finally, we will work with the microstructures metrics tool. You can select between amplitude and phase depending on what image you want to analyze. Then insert min and max area. From here, several subtools become able. First, we are going to select count particles and then click apply. The software first shows the histogram with the automatic selection threshold. Then it displays the binary set image, separating the object from the background. Then a dialog appears asking for sample Polaroid. After segmentation, the software detects connected components representing the cells. Each valid object is labeled and outlined in red. Now select the particle areas. This mode calculates the area of each detected object, which is useful when analyzing cell size variation or particle distributions. As before, a Polaroid confirmation dialog appears. After selecting the correct Polaroid, the software generates a labeled image of detected particles and displays a histogram of particle size distribution. A results table is also created, listing each particle ID, centroid coordinates, measurement area, and estimated diameter. These values are expressed in square micrometers. Next, we move to the third option, automatic face profile. This mode extracts a face profile automatically for each detected object. The software again segments the image and confirms Polaroid. Then, for each valid particle, it places a measurement line across the object and calculates the face profile. A summary window appears showing the phase difference value for every detected particle, allowing batch analysis across a population of cells.
Now we are in the Speckle section. Here you can make speckle measurements and use filters to reduce speckle. Let me show you how it works. For speckle measurements, first choose which image you want to measure, We will use the face image in this example. Number of zones means how many places in the image you want to measure. Each zone is split into rows and columns to get a more reliable statistical value from that one zone. And for this example, I will set number of zones to 2, rows to 3, and columns to 2. Then click apply. So now select the 2 zones on the image and a results table will appear with the measurements for each region. You can see the raw value not normalized, the normalized value, and the average across the selected areas. Now the speckle filters we have are HMF, mean filter, median filter, Gaussian filter, and SPP filter. You can apply them either to amplitude or to phase. First, pick amplitude or phase, then choose one filter, set its value, and press apply. Only one speckle filter can be active at a time. The first one, HMF, means hybrid median mean. It runs the filter several times and averages the result, which reduces speckle while keeping edges fairly sharp. Here you enter iterations, and iterations is how many passes the algorithm makes. More passes mean stronger smoothing. So we will first set 3 iterations, and now we will set 7. So we can see the difference. Mean filter does simple averaging inside a square window. Here you enter kernel, and kernel is the window size in pixels. A bigger kernel makes the image smoother but also makes the edges less sharp. Median filter replaces each pixel with the median value inside a square window, which reduces granular speckle and preserves edges better than averaging. Here you also enter a kernel, use an odd number of at least 3. Larger odd numbers increase smoothing. Now Gaussian filter blurs the image with Gaussian curve given a smoothing. Here you enter sigma. Sigma is the blur radius. Higher sigma means more smoothing and more blur. And the last one, SPP filter, means stochastic phase processing. It works on the complex field, so make sure you have reconstructed the complex image first. Here you enter iterations, the number of averaging steps, and more iterations reduce speckle more. And as we have seen, if you want to undo a filter, just uncheck it and press apply again. After you apply any speckle filter, you can open the comparison tool at the button, side by side, and profile. And a speckle plot only works for SPP and HMF. So side by side shows the original image and the filter image next to each other. This allows you to see how much speckle was removed and how much details was preserved. The speckle plot makes a quantitative plot for SPP and HMF. So first you select a small region in the image. Then the tool computes the speckle metric in that region for each iteration and draws a curve of normalized speckle contrast versus iterations. Lower values means less speckle. Profile boils a one-dimensional compression across a selected patch. You draw a small box. The tool zooms the pixel values in the vertical directions inside the box and plots pixels positions on the horizontal axis versus the vertical zoom on the vertical axis. You get two curves, one for the original and one for the filter imaging. So you can see how edges and fine structures change after filtering, and data with the exact values is also shown. And that's all for the speckle section.